Hi, I'm Alison Knoop, and today we're going to be looking at two very interesting examples of organisms in fungi and protists. In these lessons, we are going to focus on summarizing what you need to know to be able to achieve in your end-of-year exam. So we'll cover, for protistans, their basic structure and their general characteristics, how they are important economically as well as ecologically, as well as diseases that may be caused by protistans. And when we look at the fungi section, we will look at their basic structure as well as general characteristics, again, economic use and diseases. Let's start with the protistans. Protistans, to me, are possibly the most interesting group of organisms. They are quite an advance on bacteria in that all protistans show what we call a eukaryotic nucleus. Now, a eukaryotic nucleus is the typical nucleus structure that we have learnt about, where we have a nuclear membrane, a nucleolus, and our chromatin material or chromosomes contained within that. Protists, however, are much more advanced than bacteria. They seem to be split into almost two distinct groups contained under the heading protists, in that some of the protists tend to show more plant-like characteristics. Others of the protists tend to show more animal-like features. In a sense, you could describe these as your pre-plants and your pre-animals. They're not complicated or large enough to be true plants or animals, but they are starting to show plant or animal-like characteristics. Thus, we'll start our study of protistans by looking at some different examples. We start with terminology for the section, and again I will come back to the terminology later on. Here are some different examples of protistans. The first two pictures I've included show those that are more plant-like. If you look at these two pictures, you can see that both of them are green, therefore they have chlorophyll. That is a plant-like characteristic. If you look at the second picture over here, you can see a very clear cell wall. And what is very interesting about this particular picture, the strange ribbon-like structure that you can see spiraling through the cell is in fact a chloroplast. I think we can all agree that these examples of protistans are definitely more plant-like. However, within the same group, we have some organisms that look totally different to this. Let's look at two examples. These two examples are more animal-like protistans. The first one is one that we are going to look at in a little more detail in a moment. That is amoeba. This second one over here is an organism called a paramecium. Now, if you look at these two pictures, you can see that this amoeba does not have a regular shape. It doesn't have a regular shape because it has no cell wall. Also, if you look at the pictures, you can see that neither one of them is actually green. That's because these more animal-like protists have no chlorophyll and they therefore cannot make their own food. Remember that the correct term for an animal or an organism that cannot make its own food is heterotrophic. That means different feeders. Thus, in having to feed in a different way, you will find that our more animal-like protists may either go out like the amoeba, and hunt down and catch its food, which we'll look at shortly, in other words, it is a predator, or some protistans that are more animal-like are parasitic in nature and take their food from a host. And we'll look at a very interesting example of such a protistan a little later. Let's look at our example of the amoeba in a little more detail. Here is a schematic diagram to show you what an amoeba looks like structurally. Let's examine this diagram. First of all, you can see at part F, there is our properly developed nucleus. This, remember, is a eukaryotic nucleus because you can clearly see the nuclear membrane around the nucleus. You will also note that there are other organelles present. For example, this structure over here, indicated by G, is the structure called the mitochondrion. Remember that the mitochondrion's job is cellular respiration. It is going to provide energy for the cell to do its various activities. Other structures, here at C is a very, very large vacuole. This kind of vacuole is incredibly important. You will find amoeba living in water. Because of the difference in water potential between the amoeba and the outside water, what constantly happens is water flows into the cell. This water is gathered into the contractile vacuole, pushed towards the side, and the water is spat out. That prevents the amoeba from swelling and swelling and eventually bursting with having taken too much water in. Other things that you can see, here we have food vacuoles, and in here food is being digested away. What, however, is particularly interesting about the amoeba is its cytoplasm and its cell membrane. Even though this amoeba is only one little cell, it uses its cytoplasm and its cell membrane to enable it to move and to hunt down food. Let's look at how that happens. Here, inside the amoeba, you can see that the dots are much, much closer together and far more dense than the outside area. That's because the cytoplasm is almost in two different consistencies or textures. Here on the outside, we have a substance called ectoplasm. It just means outside cytoplasm. This is not very granular, and it tends to be more solid. The cytoplasm on the inside is called endoplasm. You can see that the endoplasm is very, very granular, and this more granular endoplasm tends to be more liquid. Now, what is interesting? The outer layer, or ectoplasm, holds the shape of the animal. The inner layer, or endoplasm, actually circulates around, and as it circulates around, so it distributes nutrients throughout the whole cell. But that very self-same principle can be used to help this amoeba move. What will happen? If the amoeba decides to move in a certain direction, it will turn the ectoplasm in an area into a more liquid state. Because it's more liquid over here, and this endoplasm is circulating and circulating, it starts to flow into this weaker area and create a bulge. The bulge will get bigger and bigger and bigger. When the bulge reaches a certain length, what will happen is this ectoplasm will be turned solid again, creating almost an anchor. The rest of the amoeba's body will then be pulled up to where this foot has been stuck out. Now, wonderful, wonderful term. These protrusions here are nicknamed false feet. And the correct biological term for those, pseudo, meaning false, podia, meaning feet. And these pseudopodia can be used to help the amoeba creep along slowly. Or, when it gets to its prey, what it can do is make two pseudopodia, stretch them out and encircle the prey, and then pull the prey into the body. So you can see that with the cytoplasm and the cell membrane, this amoeba can move and can take in food. So that then is our generalized structure of the amoeba. Let's move on. To review, general characteristics of protistans. Included in this kingdom, we are going to have some that are more plant-like, green algae, brown algae, red algae, diatoms, and slime molds, and then a large group that we call protozoans. If you look at the word, zoa is the plural. Zoo, what do you find in a zoo? Animals. So wherever you see zoa in a word, it's referring to animals. And these protozoans are the more animal-like ones, like the amoeba that we've just looked at. They are eukaryotic. They are usually unicellular. And as mentioned with the amoeba, most of them are aquatic or they live in water. Other features? Some of them, the more plant-like ones, are autotrophic, self-feeding. Others, the more animal-like ones, heterotrophic, different feeding. Remember, as I mentioned to you, that some of these different feeders are parasitic, while others, like our amoeba, and in fact the slime mold, stretch out those arms and hug in, pull in their food, which is called phagocytosis, a beautiful word which means cell eating. Further characteristics, some reproduce asexually by the process called binary fission. Binary fission is simply a copying of genetic material or chromosomes and then splitting in two. However, not all protistans will reproduce this way all of the time. Some of them have quite complex life cycles, and in fact there are those that reproduce sexually and asexually as well. So you can see that there are quite a variety of different features within our protistan group. Looking specifically at our amoeba, remember that it is unicellular. A word that we use to describe it is asymmetrical. Now, asymmetrical is that it is never really in the same shape. Because it's constantly making those false feet, it changes its shape all the time. As a result, we describe it as asymmetrical. It is always surrounded by a cell membrane, and remember, differentially or semi-permeable membrane, because this is what is going to allow the water to flow in, that it has to use that special vacuole to get rid of. The cytoplasm has ectoplasm and endoplasm. There's a nucleus for controlling all of the different activities. The special vacuole called the contractile vacuole to get rid of water, as mentioned. And we find food vacuoles for digestion and the distribution. And remember that that cytoplasm is constantly circulating, distributing nutrients throughout the whole cell. So although the amoeba is only made of one cell, there's an awful lot going on inside there. Let's look now at, generally speaking, how our protistans are important. And we're going to start with the ecological importance, because protistans are very important to us and to all other organisms on Earth. Ecologically speaking, The microscopic organisms in the upper layers of the water are called plankton. Those protistans that are more plant-like and photosynthesize are also part of plankton, the unicellular plankton. Here, they are producers in water, and often you will actually hear plankton referred to as the grass of the seas because there are so many aquatic organisms that feed on them. But that's not all that they do. You will find that autotrophic protists, and especially here those plankton on the top of bodies of water, they are going to take in carbon dioxide. Now that carbon dioxide is always taken in for the process of photosynthesis. So that's very important because our autotrophic protistans are reducing our carbon dioxide levels. And as a byproduct of photosynthesis, our autotrophic protistans are releasing oxygen. This is a very, very important process. By means of their photosynthesizing, autotrophic protistans are playing a very important role in keeping our oxygen and carbon dioxide levels balanced. Effectively, they are providing oxygen for us. Also, nutrient recycling. You will find that some of the more animal-like protistans feed off dead or decaying organic matter. Those that feed in this way are called saprophytes. Why are these good? When a saprophyte feeds on dead or decaying organic matter, it takes its own food from that matter but returns some of the nutrients back to the soil. So that's very useful as well in keeping soil and water rich in minerals. Let's look at the economic uses of protistans. First of all, silica. There is a very, very special kind of protestan that you find in marine environments in the sea called a diatom. Look at the word di, two, atom, parts. And literally these look sort of like boxes. In their cell walls, you will find a lot of silica. Silica is used as sandstone, which is used in industry. But what is quite interesting about these diatoms is many, many years ago, before plastics were so common, diatoms were actually put into toothpastes, and they gave the toothpaste that little bit of an abrasive or rubbing quality to help get your teeth really, really clean. Also, there is an interesting phenomenon called a red tide. Red tide involves algae, and what they do is they give off toxins. These toxins are then taken in by a lot of other aquatic animals. This will include shellfish and fish. Now we need to be very careful of these, because what will happen is if we eat those that have been affected by a red tide, it can cause cell membranes and of nerves and muscle cells to be affected, and it causes paralysis and even death. Finally, seaweeds. Seaweeds are used in our new most popular form of food. They are used in sushi to wrap up the various components. However, you find them in thickening powders in puddings, you find them in soups, in salad dressings, and you will even find seaweed in ice cream. Look on the ingredients on some of your household products. Any time that you see the word emulsifiers, they've probably put seaweed in there. Let's move on with our protistans. A very, very interesting example of a protestan that we are going to look at is one that causes a disease. Now, this disease-causing organism is a parasitic protestan. Now, this parasitic protistin causes the disease called malaria. Malaria is not the name of the actual organism, it's the name of the disease, and it comes from old African words meaning sick air, because originally it was thought that malaria was caused by incredibly humid, almost poisoned air. However, this has since been proven not to be the case, as it's a microscopic little parasite that we're going to look at now. Malaria is caused by a special kind of protozoan. Again, look at the word. There's the zoa, so it's one of the ones that is more animal-like. This protozoan is called Plasmodium. There are a number of different types of Plasmodium, so we're going to generalize a little bit. Plasmodium is carried by a very, very special and specific kind of mosquito called the Anopheles mosquito, and I'll show you what those look like in a second. However, not all Anopheles mosquitoes spread this disease. It's actually only the females. Male Anopheles mosquitoes are herbivores, so they are busy sucking out plant sap. Female Anopheles mosquitoes are the only ones that will suck blood, and they need something in the blood to be able to manufacture their eggs. So remember that the female of the species is always more deadly than the male. What will happen to you if you get malaria? Some of the symptoms that you will encounter include headache, very bad headache, lasting for an extremely long period of time. You will have fever, sweating, and chills, but these come in cycles, and we'll look at why in a moment. Muscle pains in your back and limbs, abdominal pains, which is a really bad stomach ache. You will have diarrhea, nausea, vomiting, loss of appetite, and sometimes even a cough. So there's quite a long list of symptoms. But what we need to remember, if you look at the first few, the headache, fever, sweating, and chills, and so on, doesn't that seem a bit like flu? We'll come back to that. It is always better to prevent malaria. Because the Anopheles mosquito tends to like to live in humid areas where there's lots of water around and it's nice and hot, there are certain basic measures that you can take in order to prevent yourself being bitten by an Anopheles mosquito. Firstly, try and get rid of them. How would you do that? Use insecticides. Or if you have open water outside, a little body, a puddle that doesn't ever dry up, a pond that you've emptied that has no drainage, so there's a bit of water that's gathered from rain, get rid of it or put oil on top of it. Oil prevents the little eggs from hatching and developing further. Other preventative measures. Try and prevent the mosquito from biting us. There is a whole range of sprays that are available that you just spray on your skin, and they smell really bad to mosquitoes, so they won't come near. You can even nowadays get them as an armband that you just wear around your arm, and that also it has exactly the same effect. Or spray insecticide around you. Taking medicines before you enter a high-risk area. Now this is incredibly important. Should you decide to go further north during the summer season, you should go to your doctor first and ask for preventative medicine. Preventative medicine for malaria is called prophylaxis. There are a number of different types of these pills available, and some of them you will need to start a few weeks before you go on holiday. Some are every day. Ask your doctor for the best kind. There are, however, other more practical measures that you should put into play as well. What, for example, about mosquito nets? What about wearing long clothing? And by that, I don't necessarily mean warm, thick clothing, but just cover your skin with a long sleeve top that is relatively thin material-wise. So there are a number of things that we can do to prevent the Anopheles mosquito from biting us. Because once she's bitten us and injected those parasites into our bloodstream, it's quite a difficult job to get rid of the disease. You will find that a chemical test called the rapid test is used to diagnose a person who has malaria. This is quite important because remember those flu-like symptoms. As well, a blood sample can be taken and you can look for the parasites in it. I will show you what those look like in the blood cells in a moment. Or what you'll find is treatment can occur with quinine, ACT. These are different medications, and it depends on which country you're in as to which is recommended. So there are a number of methods of treatment that are available to you. Let's look at the malaria parasite's life cycle. The life cycle of the malaria parasite is a particularly interesting one because this malaria parasite, or Plasmodium, uses two hosts. Host number one is the female Anopheles mosquito, and it is in this host that the parasite spends its adult phase. It's interesting how, although the Anopheles mosquito has the parasite, it has absolutely no ill effects whatsoever. The second host is man, and in man, this is where the parasite spends its larval stage. And man, as a host, is the one who is harmed by this parasite. Let's follow the life cycle and see what happens at each stage. A female Anopheles mosquito has the parasites in an immature form called a sporozoite in her salivary glands. She will use her long mouthpart to pierce the skin of a human. And what is really interesting is that she will inject her saliva into the human skin. This saliva contains, number one, an anesthetic. So that's why you don't feel a mosquito bite immediately. But number two, an anticoagulant. An anticoagulant prevents the blood from clotting whilst she's busy sucking it out. The blood is then sucked into the female mosquito, but by a separate tube. So the blood and the saliva don't travel in the same tube, they don't mix. However, this adult female Anopheles mosquito had the parasite in her salivary glands. So as she injects saliva into the human's body, so she injects the immature sporozoites into the bloodstream. These sporozoites will travel to the liver, and in the human liver they will burrow into the liver cells, use the liver cells and burst out of them. Once they burst out, they re-enter the bloodstream, and when they re-enter the bloodstream, they look for our red blood cells. Each of these new little forms, called a merozoite, burrows into a blood cell. One merozoite per blood cell. And once they're in that form, they will feed on the inside of the red blood cell and get bigger and bigger. And when they get to their largest possible size, they divide and burst out of the red blood cell, releasing more merozoites. As they burst out, so they release toxins, and the toxins cause the person with malaria to have chills and fever and all of the negative symptoms. What will then happen? Each of these new little merozoites will find a new red blood cell. Each one will burrow into one red blood cell, feed on the contents of the blood cell and grow and divide, burst out. And what is particularly interesting is the bursting out happens at regular time periods. Depending on which variety of the Plasmodium parasite you have managed to get, you can have 24-hour cycles. In other words, they are bursting out of the red blood cells and releasing toxins every 24 hours, or it can go as long as 72 hours, that every 72 hours you will have all of the symptoms. Sometimes what will happen, some of these merozoites, instead of finding a new red blood cell, will develop into a special form called a gametocyte. Now look at the word gameto, gamete cells. And these will circulate in your bloodstream and they don't do anything to harm you at all. In the meantime, the merozoites are burrowing in, reproducing, bursting out, burrowing in, but these gametocytes just go around in your blood, circulating through your body. Until another female Anopheles mosquito comes along, uses her long mouth parts to pierce your skin. She injects her saliva, and as she sucks out your blood, she will suck out these gametocytes with it. The gametocytes travel through the mosquito's gut and reproduce. They migrate back to the mosquito's salivary glands and form the form called the sporozoite, and they will lie there in the mosquito's salivary glands waiting for her to find her next blood meal. Kind of scary, hey? Let's look at some pictures. Here is the female Anopheles mosquito in flight. She is often nicknamed the silent killer. The reason for this: the mosquitoes in Gauteng, you can hear them buzzing around your room all night. However, if one of them is an Anopheles mosquito, you won't hear a thing because they do not make that particular high-pitched whining noise. And what is really interesting about this picture, look at her red abdomen, which is engorged with blood. Our next picture shows a female Anopheles mosquito on the skin using her mouth parts to pierce the skin. There you can see there, the mouth parts are getting ready to pierce. Here is the form that she has injected into your bloodstream. This is the sporozoite that is traveling through the bloodstream, and remember, it's going to go to the liver, where it's going to use the liver cells to reproduce. Here is our next picture. Those sporozoites reproduced in the liver cells. They have made the special little form called merozoites that went out into the bloodstream. Now look at what happens to the merozoites. Here, each merozoite will find its own red blood cell, remember, one per blood cell, and burrow in, and these will start to feed on the blood cell and get bigger and bigger and bigger. Now look at the second picture over here. Look at what happens to those blood cells. Can you see that they've gone sort of pear-shaped? In other words, the blood cells are actually physically deformed by these growing merozoites. Now, this is one of the things that they'll look for when they take blood from you to diagnose if you have malaria or not. Now remember, here they're getting bigger and bigger and bigger. They're going to divide and they're going to burst out of the red blood cell. And here is the most beautiful picture. Look at those red blood cells and look at those little parasites bursting out. Each one of these little balls is one of those Plasmodium parasites, and each one of those is going to look for a new red blood cell to infect. Remember, as this bursting happens, so toxins are released. What is the toxin going to cause? The cycles of chills, fever, sweats. the headache, etc. So here, this picture actually shows you when this is going to happen. Let's move on. Here are the gametocytes, and these gametocytes, remember, are the ones that are just going to be in your bloodstream, circulating around and doing absolutely nothing to you. Of course, whilst they are circulating, remember that those are reinfecting and bursting out. And this is the most exquisite picture. Here comes our female Anopheles mosquito. She is going to suck up those gametocytes into her gut, where they will reproduce, form the sporozoites that will lie in her salivary glands. And look at the length of that mouth part, and yes, that really is what human skin looks like close up. So there's the life cycle of the malaria parasite. Really, really very interesting, and a huge cause for concern for all of us living in Africa, because this is where you find most new cases of malaria reported every year. Let's move on. We're now going to look at some examples of different questions that you may be asked on this section. Question 1 deals with the amoeba. We're given a diagram and asked a number of questions based on this diagram. First question, identify each of the following. Firstly, outermost layer E, there it is. Okay, from what I can see, they're actually pointing to the line there. So E is going to be the cell membrane, or remember the correct term for it, the plasmalemma. Gel layer, labeled B, there it is over there. That's the cytoplasm on the outside, correctly called ectoplasm. And then the organelle of respiration, there's our clue word, labeled G. There it is over there. What is our organelle of respiration? The mitochondrion. And finally, the organelle of osmoregulation, designed to confuse you, labeled C. Look at the word. Osmosis, always is going to refer to water. Which structure in this diagram is responsible for regulating water? Answer, here, bails water out. What is its correct name? It's called a contractile vacuole. Let's move to question 2. Question 2 gives us a reading study, and remember to highlight important bits as you go through. Malaria is a sporozoan and a very important human parasite. World Health Organization reported that more than 3 million people died of malaria in 2003. Malaria parasites are transmitted from one person to another by the female mosquitoes of the Anopheles species, which acts as the vector. Vector is just the way that something is spread. People can stop the infection by sleeping under nets, use repellent lotions, insecticides, smoking herbs in the room, spray oil on stagnant water in pots and old tires, and cover the bodies with protective clothing. Now, let's look at the kinds of questions. First of all, which organism causes malaria? Now be very careful when you answer this, because if you look at the information given in the reading study, the only organism that is named is the Anopheles mosquito. Please remember that is not the organism that causes malaria, it is the one that transfers it from one to another. The actual organism is called Plasmodium. The next part of the question. Explain 2 effects of malaria on the human body. Well, here there is quite a list, So any two of these. You may have cycles of chills and fever. You may have headaches, nausea and vomiting. You may have muscle pains, and so on. Remember that a whole list of those was given in the table where we discussed the disease. So literally any two from that table will get you your marks. The next part of the question deals with two environmental conditions beneficial for spreading malaria. In other words, because malaria is spread by the mosquito, what environmental conditions are actually good for mosquitoes to reproduce in? And we've done those as well. Remember, it must be humid, it must be hot, and preferably there should be some kind of water body around for reproduction. The next part of the question, one of the symptoms of malaria is fever. Why is it that malaria patients break out into a fever every 2 to 4 days? And again, we've done that. Remember that it's every 2 to 4 days depending on which specific plasmodium you are infected with. What you would need to say here. Fever only occurs when the merozoites burst out of the red blood cells. This happens only every 2 to 4 days and releases toxins. What do those toxins cause? Those cause the symptoms, which, as we've discussed, the headaches, the chills, etc. Next question. Name 3 methods that form the basis for the treatment of malaria. This question also was dealt with in the table. So let's look at some of the possible answers here. The first one that was mentioned was using quinine. Then there were 2 different types of medications mentioned. You will not be expected to know the names of the medications, just that there are 2 types and that you could say that they are country specific. So it's going to depend on which country you're in as to which of the 2 you take. Also, because you're going to be having headaches, because you're going to be having muscle pains and so on, I would say that the third point that you could put in here is treatment of symptoms and that would include things like headache pills. And please note it says the treatment of malaria here, not the prevention. However, prevention is one that is very, very commonly asked. So please make sure that you know how to prevent malaria as well. Final part of the question. Indicate 2 reasons why malaria is not easily diagnosed. The first part of this question we mentioned when we looked at the symptoms in the table. First of all, it is very, very, very similar to flu. So often malaria is initially confused for a flu infection. So the 2 are very confusing. However, our second point, if a doctor does a blood test and sends that blood test off to the laboratory, they are going to make a slide with a sample of blood on it. If the merozoites have just gone into the red blood cells and they are still small, then you cannot see anything in the blood and you can't actually see anything in the blood cells. So it's very, very difficult to detect in the blood unless those merozoites have filled up the blood cell, and remember, given it that particular pear shape, or they have burst out and they're actually in the liquid part of the blood. So we'll just say that it's difficult to detect in the blood. Thus here, confusing and difficult will give you your 2 marks. Now that we've done the content, let's go back and revise the terminology. Aquatic is a feature shown by most of our protists. Most of our protists live in or around water. This is because the majority of them will use water as a medium in which to move, as well as a method of body support. Our second term is eukaryote, or an organism may be described as eukaryotic. Remember that eukaryotic means that it has a definite membrane around the nucleus, so that there is our nuclear membrane, there is the nucleolus, and the nucleic acids are inside. Make sure that you are able to compare eukaryotic or eukaryote with a term done in previous chapters, that is prokaryotic. A prokaryotic nucleus is one where there is no clear membrane, just the nuclear material, as is shown in our viruses and bacteria. Unicellular means only made of one cell. Asymmetrical is a characteristic shown by amoeba, and remember that asymmetrical describes it because it constantly changes its shape. Now let's start to look at fungi. You know how every family has that strange cousin? Well, fungi are the strange cousins for all other organisms. They look sort of like plants, but they are not plants. They have certain animal-like characteristics, but they're not animals either. So what are they? They are fungi. Let's be a little more specific. All fungi have eukaryotic nuclei, so they have properly developed nuclear membranes around their nucleus. They are like plants in that they all have cell walls, but no fungi will ever have chlorophyll. Now that is more of an animal-like characteristic. Because they don't have chlorophyll, we call them heterotrophic. And remember that the word heterotrophic means different feeders. So how do fungi obtain their food? Normally it's in one of 2 ways. They can either be saprophytes, and remember that saprophytes feed on dead and decaying organic matter, or fungi may be parasites. Parasites are organisms that will use a host for food and therefore will damage that host. Because they damage the host, these fungi will cause disease, and we thus call them pathogens. There are a number of different examples of fungi. Some of them you may be familiar with. Here we have a mushroom type of fungus that you can see growing on an old tree stump. This kind of fungus is a saprophyte because it's breaking that dead tree stump down and returning the nutrients to the soil. This picture shows a particularly beautiful mushroom. Be careful of the mushrooms and toadstools that are brightly coloured. Those are generally the poisonous ones. Here is another example of a mushroom type of fungus. Further examples will include one that we may use at home for baking. Another example then, yeast. It's a fungus. But the one that we are going to study, I'm quite sure that all of you will have encountered it in that lunchbox that you forgot in your case over the school holidays with your half-eaten cheese sandwich still inside. What am I talking about? The wonderful bread mold. The correct name for bread mold is Rhizopus. And you can see that there are many, many different colors as there are many different types growing on this piece of bread. Literally, this bread mold is growing on its food and it is secreting digestive enzymes into the bread, breaking the bread down and then sucking it up through its roots. So it's obtaining its nutrition in a saprophytic manner. Let's move on to look a little more specifically at the structure of bread mold or Rhizopus. Here you can see a picture of bread mold, a schematic diagram. First of all, when a bread mold starts to grow, you will see a tangle of fine threads on the surface of a piece of bread. Each of these fine threads is called a hypha. Hypha is the singular. To make it a plural, just add an E on the end. As these hyphae start to spread across the bread, you will see that they grow in 3 different directions. First of all, hyphae will grow down. The ones that grow down penetrate the bread, and they are the ones that are holding the bread mold in place as well as digesting the bread away. These are root-like structures, so we call them rhizoids. They are not true roots because they are not absorbing water only, they're absorbing food as well. They also don't have the structure of a proper root. The second kind of hypha grows across the surface of the bread, allowing the bread mold to grow further and further over the piece of bread. These ones that grow sideways are called stolons, and they allow the bread mold to spread. The final kind of hypha that will grow is one that grows upwards, and this kind of hypha is called a sporangiophore. Please be careful of the spelling of that word. A sporangiophore is a stalk that will hold a sporangium eventually. So those are your 3 kinds of hyphae growing in all possible directions: rhizoids growing down, stolons spreading across, and the sporangiophores growing up. Note that all together we use the term mycelium, which is a very fungal sounding word to describe all of those different threads all together. At a certain point, when the sporangiophores have grown high up enough, what you will find developing at the top are little sacs. And these little sacs are called, here's one, sporangia. One sporangium, plural, sporangia. These little sacs contain the spores. And what will happen, the wall here, you can see it, it's called the columella, will start, as you see here, to move up and further up into the sporangium. This will create the most incredible pressure buildup on the inside of the sporangium, so that sporangium will burst open, and as it bursts open, so these tiny little structures, which are the spores, will be flung out. They are flung out to take them as far away as possible from the parent plant, so that they can grow on their own little piece of bread and not compete with the parent plant for food. So here, my basic bread mold structure. We have our rhizoids at the bottom. There you can see our stolons growing across the surface, and here are the sporangiophores. All of these together is the mycelium, all of the fungal threads, and at the top there's your sporangium filled with spores. That label line there to the columella, and there you can see it bursting and flinging the spores out. So, quite an interesting little structure. Let's move on. The general characteristics of our fungi, such as those shown by mushrooms, yeast, and the molds. First of all, because they have cell walls, they are sort of like plants. However, unlike plants, they don't have chlorophyll, and thus they must be heterotrophic. When they are heterotrophic, they may be parasites or saprophytes. Most are multicellular, but some are only one cell, and they reproduce by means of spores. So we move on. Looking specifically at our bread mold. Bread mold, such as Rhizopus, is described as being a thallus, and this is quite an important term. A thallus is any structure that has no true roots, stems, or leaves. So in other words, although my Rhizopus or bread mold is sort of plant-like in looks, those rhizoids, the stolons, and the sporangiophores are not proper plant structures, so we say that the whole plant body is a thallus. It is made up of threads called hyphae. All of the threads form the mycelium, and remember that's the down ones, the across ones, and the up ones. Remember to know what each of those are called. Then they are cenocytic, as the cytoplasm and nuclei are distributed throughout the hyphae. This is quite a complex concept. I'm going to mention the next point, and then I'm going to go back to this. No cross walls. Now, this is a particularly interesting point that makes fungi so unusual. A moment ago I said that some of them are multicellular and some, like yeast, are unicellular. But in actual fact, fungi are both unicellular and multicellular, which sounds a little bit strange. Let me explain. If we take, there is a very schematically drawn fungus. If we take from there just a tiny little piece of that sporangiophore and we make it very big, all right? So just that little area in there that I've really enlarged. What you will see is, first of all, on the outside, we have here our cell wall. And notice that we still call it a cell wall. Then, running through the whole inside, up to this little columella, up to the columella, all the way through here, we have one large continuous vacuole. All right? So in other words, through the whole bread mold, there is one vacuole. Then, many of these little structures, and these are nuclei. So in other words, it's got many nuclei and the correct term for that, multinucleate. But in normal plants or animals, you will have one nucleus in a little box or a cell. Here, there are no cross walls present. Because there are no cross walls present, what you have is many nuclei, but no separate little boxes. So, in other words, this fungus is multicellular. Why? It's got lots of nuclei. But the nuclei are not in their own individual little boxes or cells. There are no cross walls. Therefore, it's unicellular. Now, that is a bit of a strange condition, to be multicellular and unicellular. Why? Many nuclei makes you multicellular. No cross walls makes you unicellular. And that's why this term, cenocytic, is a special word for fungi and means that they're both multi and unicellular. Learn that word. It's an important terminology word. There are three types of hyphae, as mentioned: rhizoids that grow down, stolons that grow across, and the sporangiophores that grow up. And remember, when the bread mold becomes mature, the sporangiophores will develop sporangia on them. Fungi, despite the fact that they are so peculiar, are very, very important to us, economically and ecologically. We're going to start with their economic use. First of all, as strange as it may seem, fungi are used as part of the dairy industry. This is because certain fungi are used in making cheese. Cheese making employs molds like penicillin, Roquefort for Roquefort cheese, and Penicillium camemberti for camembert cheese. In other words, when you buy a cheese and you open that cheese up and you see the lines of blue running through it, that's a fungus growing on your cheese. It adds to the flavor. Next, fungi are incredibly important in medicine. Fungi are used to produce antibiotics. Antibiotics are there to kill bacteria. Please note that antibiotics don't kill fungi. The most important example is penicillin, and penicillin comes from the white blue fungus called Penicillium notatum. So fungi are very important to us, and the development of penicillin was one of the most incredibly important advances in medicine and saved millions of lives. Also, fungi are a source of food. We all eat mushrooms, and you also find quite a delicacy that they use pigs or specially trained dogs to scent out. Those are called truffles, and truffles grow under the ground near the base of certain trees, and they are quite a delicacy and they are quite expensive to purchase. Certain fungi, however, are parasitic, and parasitic fungi, because they use the host for food, will always cause disease. One of the interesting examples is ringworm. Many people think that ringworm is actually a worm growing under the skin, creating a coiled up sort of red itchy patch on the skin. But it's not. It's actually a fungus. And so too is that particularly nasty fungal disease called athlete's foot that you can get from using communal showers like at a gym without going into a shower with flip-flops. If you go to a holiday camp or if you use a gym, rather take a pair of flip-flops with you to use in the shower. But the one that we're going to look at is the very interesting example of thrush. Thrush is correctly called candidiasis. That is the disease name and the common name, thrush. What is thrush caused by? A special fungus called Candida albicans. And albicans means white, so it's literally the white fungus. This fungus can grow in the mouth and in the vagina. It can also grow through the intestines, and it can grow through the anus as well. Why might you get thrush? The most common cause is by being on antibiotic treatment for a long time. There are natural populations of Candida living in the vagina and in the mouth. Specifically in the vagina, however, there is also a population of bacteria, and the fungal population and the bacterial population keep each other in check. When you've been on a course of antibiotics, what happens is the antibiotics kill off your bacterial population, so there's nothing left to control the population of fungus. So what happens is that little Candida albicans overgrows. And when it overgrows, that is when you get thrush or candidiasis. Let's look at some of the symptoms. Specifically for vaginal candidiasis, it is the most incredibly itchy sensation, which is accompanied by a creamy discharge from the vagina as well. Generally, you will also feel quite tired, quite run down as well. You will feel burning during urination or during sexual intercourse. Now, vaginal candidiasis, obviously, is restricted to females. However, it can be passed from a female to a male or vice versa. If a male gets it, what you'll find is an irritation, redness, burning, itching on the tip of the penis, or it can occur under the foreskin. This itching and general discomfort is also accompanied by a slight discharge, and in both cases, the male and the female, the discharge does have a smell to it. The male will also experience discomfort when urinating. Babies can get two kinds of thrush, or thrush in two specific areas. First of all, they can get it on their bottoms, around the anus, and it looks like a really, really bad nappy rash. Obviously, it's going to be itchy and burny and very uncomfortable, and a small baby can't do anything about that itchiness. However, babies are far more prone to something called oral thrush, where if you look inside the baby's mouth, on the inside of the cheeks and sometimes on the tongue, what you'll see are white patches of fluff. They have oral thrush. Oral thrush, however, is also one of the components of AIDS, of the syndrome of diseases that can be caused by advanced HIV infection. With babies, you need to be very careful about sterilizing their bottle teats very thoroughly. If these are not properly sterilized, then the Candida spores will be on the bottle, which they will then put in their mouth, and that will cause the oral thrush to develop. With prevention, the best method of prevention is to eat yogurt, but specifically when you buy yogurt, if you're on a course of antibiotics, make sure that you look carefully on the container of the yogurt cup. Make sure that it says AB cultures or contains live cultures. That means that there are live bacteria in the yogurt that you're eating. These bacteria will then repopulate the population of bacteria that has been killed by the antibiotics. However, you can also get something called a probiotic. A probiotic is normally a chewable pill that also contains live bacteria. Probiotics are very, very good, but be careful. If you're on antibiotics, you cannot wake up in the morning, take your antibiotic and your probiotic at the same time, because the medication in the antibiotic will kill all of the live bacteria in the probiotic. So wait 4 hours between your antibiotic and your probiotic pill. Candidiasis, or thrush, is actually relatively easy to treat, if not pleasant. You can use antifungal drugs. Antifungal drugs are special drugs that are designed to kill only fungi. Antibiotics don't work on fungi. You can help yourself as well by wearing loose cotton clothes and underwear, and preferably not G-strings, because that actually spreads the infection further. Note that the antifungal drugs can also be included with something called a pessary. A pessary is an antifungal cream that is actually inserted into the vagina. This also does come in the form of a pill, and it kills the fungus actually in the vagina, so you can experience relatively quick relief from thrush. With thrush, however, please try and make sure that most of the underwear that you wear is cotton, or that it has a cotton gusset if you are a girl. Also, again, try and avoid wearing G-strings. Let's move on. We're now going to look at some examples of questions that you may be asked on fungi. The first question shows a diagram of a transverse section through a potato leaf, which is infected with potato blight. Now, potato blight is a particularly nasty fungus that, if it gets into a potato crop, can make that crop completely unusable. Here is the diagram and our first question. Supply labels for parts 1, 2, and 3. If you look at label number 1, it's actually pointing to these 2 cells, or this area over here. Thus, what you can see, the potato leaf, there you can see in the darker color, that's the fungus growing out of it. So label number 1 over here is going to either be guard cells or stomata. Label number 2 is pointing to the stalk, and if you look at the stalk, it's got sporangia at the end. Therefore, label number 2 must be a sporangiophore. And as mentioned, you can see there how it's bursting open. Because this isn't particularly clear as to where the label line is pointing, rather cover yourself and give 2 options in this case. By this I mean the label line could be pointing to the dots, or it could be pointing to that structure. Therefore, cover yourself by saying spores in sporangium. Then whichever answer is in the memo, you are guaranteed of getting the mark. Thus, simply providing labels, 3 easy marks. The next question. What is the normal function of part numbered 1, which is the opening? And remember, we said the guard cells or the stomata. Now that they've said it's the opening, we know it's the stomata. Here, we will accept either transpiration, which, if you remember, was done in an earlier section of the syllabus with water movement through plants, or you could here also answer gaseous exchange, in the sense that these are the special openings on the leaf where CO2 is going to move in during the day for photosynthesis, and O2 is going to move out because it's a waste product. So either one of those 2 options will get you the mark. The next question, to which group of microorganisms is potato blight classified? So which group that we've studied does this fit into? It is a fungus, so it fits into the group called fungi. Simple, 1 mark. Still with the same diagram, explain why potato blight usually infects plants growing in shady areas. In shady areas, remember that it is cool, that it is dark, and that it is also moist, in that there's plenty of water in the shade. As a result, these 3 conditions are all very, very, very favorable conditions for fungal growth. Remember, that's why your cheese sandwich grew that fluff on it in your lunchbox. So fungi's favorite conditions are cool, dark, moist areas. Next question. One way in which this disease can be controlled. There are a number of ways that fungal diseases can be controlled. First of all, you can spray your crops, and you can spray them with an antifungal agent. By spraying them with an antifungal agent, you should kill spores that are forming on the potatoes themselves or on the potato plants. However, you could ensure that they should be grown in areas where it is light, where it is warm, and this won't affect the potato part of the plant because the potato is underground. So light and warm, that will help counteract any fungus that might want to start growing. And as well, what you could do here is make sure that your plants are properly spaced. Why would that be an important concept? If you space them, there's far less likelihood of the fungal spores being able to spread from one plant to the other. Now, the question only asked for one way, so any one of those would give you 2 marks. Let's move on. Here's our next question, in which you can see a wonderful picture of our rhizopus or bread mold. First of all, identifying parts A, C, and F. There's part A, this round part at the top. What is it? It's the sporangium. Part C, how is it growing? Sideways, spreading. That's a stolon. And part F is these and these and these. So what do we call the ones that grow down, grow across, and grow up all together? They are called a mycelium, an important terminology word. The next question. Is this plant a parasite or a saprophyte? A nice easy question for us. This plant, the bread mold, is very definitely a saprophyte. Why? Because it's feeding on dead, decaying organic matter, which is bread. The next question. Give a reason. I think I just did that. You can say that bread is not living, and this then is feeding on dead, decaying matter. 2 marks, so please make sure that you give 2 points for that. 1 function of part D, and remember I mentioned that right at the beginning. You can, of course, say anchorage because it does hold the bread mold onto the bread. But more importantly, it's going to absorb digested bread. Remember, that is how the bread mold gets its nutrients, gives off the digestive enzymes, the bread is broken down, and the digested bread is sucked right back up again. 1 function, either one of those, for 2 marks. Question 3. In the early parts of the 20th century, spiders' webs in which many fungal spores were trapped were placed on small cuts and wounds. Suggest one reason for each of the following. Why this might have been a useful procedure. Remember that fungi are used to make antibiotics. As a result, the fungal spores probably would have antibiotic properties or, antibiotic properties or antiseptic properties, and therefore would serve to prevent infection. Why could it have been dangerous? Well, there may have been fungal spores in there, but not all of those fungal spores may be from harmless fungi. Some of them may be parasitic. As a result, why it could have been dangerous? Some of the spores could have been from parasitic fungi. And what would that thus result in? They may have actually caused an infection, or they might have caused a different infection. Now that we've learned about the structure of different kinds of fungi, let's go back to revise our terminology. Heterotrophs are those organisms that cannot manufacture their own food. This is because they have no chlorophyll. As a result of not having chlorophyll, all heterotrophs have to look for their food in different ways. Fungi find food in one of two ways. First of all, some of them are saprotrophic or saprophytic. This means that they get their food from dead or decaying organic matter. And remember that as they obtain their food, so they decompose the organisms on which they are growing. Other fungi may be parasitic, where they use a host to obtain their food. A thallus is any structure which does not have true roots, stems, and leaves. Remember that in a fungus there are fine threads called hyphae, which grow in 3 different directions, but we'll come to those in a moment. Another term that is important is the word mycelium. This is the mass of entangled filaments. In other words, to make that a little bit easier, a mycelium is the term that we use to describe all of the different hyphae that grow down, across, and up. What are those? The ones that grow down are rhizoids. They are root-like hyphae. Remember, they are not true roots, as their job is not only to anchor, but also to give off digestive enzymes and to absorb the bread that has been digested. The ones that grow sideways are called stolons. They creep over the surface, and as they creep over, they allow the fungus to spread out over the bread. Our third kind of hypha is the one that grows upwards. It is a long stalk which will develop a sporangium. Sporangiophores are important because they're going to hold the sporangia right high up off the surface of the bread. I'll tell you what that's going to mean in one sec. A sporangium is a spore case or spore sac. Inside the spore sac are spores. Now, as the sporangiophore holds the sporangium far up off the surface of the bread, when the sporangium pops open, those spores are flung further than if the sporangium were far lower down. So again, it's a method of the parent plant reducing competition. By throwing the spores far away, there is nothing that will grow close to it to share its food. Our next term, an antibiotic. Antibiotics are special medicines that have been developed, and the majority of them have been developed from fungi. They are very, very useful in controlling the growth of bacteria. But what is interesting, remember that an antibiotic will not work on a fungus. Should you have a fungal disease, you will need to get antifungal medication to control it. Fungi and protists are two particularly interesting groups. You can see how much more developed they are than viruses and bacteria, and how, although they seem structurally to be quite simple, they're actually pretty complicated. Remember again that these are things that we encounter every single day, but that we don't normally know that we're encountering a protistan or a fungus. That's the end of our protists and fungi section.